Hello class. So this is the second part of the series Nucleotide Biosynthesis. And so we are going to cover the topics of purine bases can be synthesized from glycine aspartate and other components. And the other topic will be deoxyribonucleotides are synthesized by the reduction of ribonucleotides. So, like pyrimidine nucleotides, purine nucleotides can be synthesized by de novo or by salvage pathways. And so this figure right here, this shows where some of the atoms that make up the purine ring structure is coming from. Where you see right here that is highlighted in yellow is from the glycine, this carbon right here, and this carbon right here as well that's highlighted in green comes from the N10 formyl tetrahydrofolate. And then you have both of these nitrogens are coming from glutamine. And then you see right here attached is the sugar and the phosphate group. And then right here for this nitrogen that is in black is from the aspartate amino acid, and this carbon that is red is coming from carbon dioxide. So with the de novo purine biosynthesis, like pyrimidine biosynthesis, it requires that PRPP, so that activated sugar by having that pyrophosphate group that is attached to the sugar. However, for purines, PRPP provides the foundation on which the base are constructed. So before with the pyrimidine, the base was formed and then it was attached to the PRPP, but now you actually have the structure coming from the PRPP in general, and it's attached to it and grows into a ring structure while it is attached to the sugar. And so for the commitment step of producing these purine is having the glutamine goes through hydrolysis, releasing free ammonia, which ends up displacing the pyrophosphate group, and you have this amine in the beta configuration for this ribose ring structure. And so further building that purine ring, so this assembly involves 9 additional steps to form this purine ring, and most of the steps will involve this type of reaction that is listed here at the bottom, where you have a carbonyl and you have a phosphorylation that's happening with ATP, where it attached a phosphoryl group, and then from there you have a nucleophile that ends up displacing that phosphate group and end up having your nucleophile attached. And so this attacking group can also be ammonia or a amine group. So in this slide, it just shows you in general all of the enzymes that are involved in the 9 steps of making your purine synthesis. So this is just something for you to reference and try to see if you can kind of figure out from memory what each step is happening within each of these enzymes in order to form the purine ring. So what's happening with the structure of producing this purine ring, which is the de novo pathway? The first step is having the carboxylate group of a glycine, which is listed right here. Residue is activated by phosphorylation using that ATP. So once again, you have phosphorylation that's happening right here, and you have a nucleophile that is displacing that phosphoryl group, and now you have this new attachment from the glycine residue. And so that is the first step. The second step involves the tetrahydrofolate, so that is THF, and then you have this, what's highlighted in blue is the 4-mil. So this whole structure is called N10 4-mil tetrahydrofolate. So it donates the 4-mil moiety to your intermediate, where it has this amine group that is there. And so it attaches. You have glutamine, goes to hydrolysis, has that free amine. That phosphoryl group is going to attach to this carbonyl and gets displaced from the free ammonia and have this final structure, and then goes through this process again of having the phosphoryl group being attached to this carbonyl, but you can have this rearrangement to end up forming a ring structure because you have this nucleophile that's right here that ends up doing displacing the phosphate group that was attached here from the ATP hydrolysis. And so now we have this imidazole ring structure that's found in purine. And then for step 5, you have, this is where the carbon dioxide is coming into play, where you have bicarbonate. It gets attached with ATP again. You have that phosphorylation that attached to this what used to be the free amine group, and gets displaced by the bicarbonate compound here, and there you have this, and then from there this product from the reaction in this step 5 goes through rearrangement to transfer the carboxylate group to the actual imidazole ring to have this final structure right here. And so we can still keep building to make your purine. And so from there, you have aspartate that comes into play. And once again, you need this ATP hydrolysis where the phosphorylation is happening right here, and it gets displaced with the aspartate amino acids and finally form this structure. And then from there, you have this cleavage of fumarate to finally have this structure. Then once again, we have the N10 4-mil tetrahydrofolate that donates the 4-mil moiety again to this a free amine group that's right here, and then you start to see like all we need now is this to close this ring, and we have the purine ring structure, and that is done by hydro dehydration, so loss of water, and then you have IMP. And so this IMP is used to end up making ATP or GTP for RNA, and then ATP can be made in different steps to end up making the dioxy version of ATP, and the same thing for GTP. So how do we get from AMP to ATP or GTP? There is two different pathways where if you involve GTP with that hydrolysis and having aspartate amino acid, uh, once again you have that's phosphorylation that happens there and gets a nucleophilic di- displacement, and that's with the amino acid aspartate to have this structure right here, and then the fumarate, that's cleavage that happens right here, and the fumarate is released, and you finally have AMP, which we know if you can add some more phosphate groups to the ribose ring, then you end up having AMP to ADP to ultimately ATP. And so if you go in the other direction with AMP to make GMP, you need NAD plus, which you know oxidize, and so from there, it forms with water this double bond carbon, double bond to oxygen right here. And then you have glutamine going through hydrolysis to provide that free ammonia that can be used as a nucleophile. And once again, using ATP as going ATP having its hydrolysis, donating that phosphate group, and it gets displaced with the ammonia, and then you have that free amine group that is now attached, and now you have GMP that can be converted into GDP, and then finally GTP. And also just take note as far as regulation. In order to make AMP, you actually have to have ATP. And so for this one, which actually end up making GTP, in order to have GTP, you have to have ATP. So these two can self-regulate with each other in order to keep producing ATP or GTP. And so as far as the salvage pathways of making a purine, um, this is done by generating free purine bases that is derived from the turnover of nucleotides or from our diets. And then from there can attach to the PRPP to form the purine nucleoside monophosphate, which is shown here, where they have the enzyme adenine phosphoribosylribosyl transferase, that is the enzyme that catalyzes the adenine to be attached to the PRPP, releasing the pyrophosphate group, and finally having your nucleoside right here. And then also notate that when you have guanine, can actually attach to PRPP to produce the GMP 'Cause I know I just showed you with IMP being used to generate GMP right here. This another opportunity for that to happen is with the salvage pathway, and that's using guanine, and from there being attached to the PRPP to produce that GMP. But also, if you have xanthine attaching to the PRPP, that actually produces IMP, which you know is a precursor that also can generate GMP and AMP. So those are all the different pathways of kind of recreating all of these different nucleotides. And so now we move on to the deoxyribonucleotides are synthesized by the reduction of ribonucleotides. So this happens in several steps where you have an electron in the first step is transferred from a cysteine residue on R1 to a tyrosine radical on R2, generating a highly reactive cysteine thiol radical, and that is right here. And right, the sulfur right here is from the cysteine residues right here, and this structure right here is coming from the tyrosine. And notate that your nucleotide or the ribonucleotide is attached to each of either the tyrosine or the cysteine of the enzyme that is used to reduce the ribonucleotide. Okay, so now that we have this radical, the radical abstracts a hydrogen atom from the carbon 3 right here of the ribose nucleus. And so now you see that radical is now on the C3, and now the cysteine residue now has a hydrogen that is attached. And so that happens for step 2. And so in step 3, the radical at the C3 releases hydroxide, and you'll see that the hydroxide is from the carbon 2 that is going to end up being released, and it actually picks up this hydrogen from the cysteine residue to ultimately release water. And so now that carbon 2 now picks up that radical. And so going into step 4, a hydride ion is transferred from a third cysteine residue. So notice this hydride right here is being transferred over. So that's why you see this happening. And that results form a disulfide bond. And so pretty much deactivating this part of the enzyme. And then from there, for going into step 5, the C3 radical recaptures the original obstructed hydrogen atom that it came from this cysteine residue. So it takes this hydrogen back, and now you have that radical back onto that cysteine residue. And then going into step 6, an electron is transferred from the R2 to reduce the radical that is right here, which will also accept a proton from there. And so now you don't have this radical that is there. And then from there, going into steps 7, now you have this reduction of that ribonucleotide, so now it can be released and it leaves this R1 position, and then from there, in order you have this disulfide bond, and this needs to be reduced, and then once it is reduced, it can begin the cycle all over again. And so, as far as a reminder, when we seen in the previous lesson, this buildup of the pyridine ring. Remember that ring is made out, not attached to the ribose sugar. It is built up outside of it and then attached to the PRPP, that ends up generating this UTP and CTP, and then we get into having that reduction of the ribonucleotide. And so just talking a little bit more into when you have the form of UDP and potentially CDP, that is all showing the breakdown of how do we get to this DTMP, where you have the ribonucleotide reductase that reduces these two ribonucleotides, and then when you have that reduction, notate from there to add in some more phosphate groups, so where it's not just di, but it goes to tri phosphate. You have the enzyme, the nucleoside diphosphate kinase. And then if you're trying to go from DCTP to DUTP, you have deaminase as an enzyme to do that. And then you have UTP ACE that ends up taking up the phosphate group, where you have instead of UTP, DUTP to DUMP, and then from there finally going into the DTMP, using this enzyme. And so highlighting how do we get to this part is you have your DUMP, and it utilizes a N5-N10 methylene tetrahydrofolate as a methyl donator onto the UDMP, to end up creating a DTP. So this is also a methyl donator. So don't confuse it with what we mentioned previously, using SAM as a methyl donator. This one is not using SAM. It's using the N5, N10 methylene tetrahydrofolate. This also ends up producing 7,8-dihydrofolate. And so in order to regenerate your methyl donator, that 7,8-dihydrofolate gets reduced with NADPH, using the enzyme dihydrofolate reductase, and you see that hydrogen is being added here. And then from there, it uses the amino acid serine, PLP, and the enzyme serine hydroxymethyltransferase, which ends up producing glycine, and also regenerates our methyl donator, the N5-N10 methylene tetrafolate, that can continue to convert the DUMP to DTMP. And that concludes the second part of this lecture of nucleotide biosynthesis.